Good morning, and welcome.
Morning, everyone. Welcome to our webinar. We'll give everybody about 1 minute or so to get settled and everybody get a chance to get in, and then we'll get started.
Excellent. So, Mark, how was the chiropractor?
It was good.
There we go. You remember that joke I told you about the chiropractor?
Which one?
It was about a week back.
Oh, yeah.
Oh man, I used to not believe in chiropractors, but then I went in for adjustment and now I stand corrected.
There we go. All right. Well, thank you everybody for joining for our webinar this morning. My name is Mark Kinsey. Just to give you a little background, we will be recording this webinar. If you have questions, please feel free to post those questions, and we will work to answer those questions at the end of the presentation. So again, my name is Mark Kinsey. I'm the U.S. Director of Sales here with Biomakers. I've spent my entire 25-year career of working in ag, from sales to working with precision technology, data science, and bringing innovation to growers to make better decisions. Scott, you want to introduce yourself?
Absolutely. I have been studying soil microbes since 2013. And when I was pursuing an agricultural science master's degree at Missouri State University, I weaseled my way into the biology department, where I got to employ next generation DNA sequencing technology to characterize the soil microbiome for my master's thesis. And I found my way back to soil genomics last year when I joined Biomakers as a technical sales rep.
Great. All right, next slide. So a little about Biomakers. This is some background. So Biomakers, our goal is to integrate microbiology into agricultural decision making to optimize farming practices. So you can see we've been in business for 10 years. A lot of work, world's largest database of microorganisms, along with, next slide. Along with some of our achievements and partners we've been working with. So just that's a little bit about Biomakers if you're not familiar. But let's dive right into why you guys joined the webinar today. And that's, under what circumstances is it advantageous to do DNA soil testing in season? So the first to answer that, let's go to the next slide, would be the most obvious is if we've got an area of the field that is performing well and an area of the field that is not performing well. So how can we use DNA testing? We're already using processes like soil sample, tissue sampling. But what other insights might be generated from being able to test a poor performing versus a better performing area of the field? There is two other options that could be done. We could be looking at trying to identify why are two areas of the field being well, or why are two areas of the field performing not so well. Again, I think one of the keys is working with at least two samples so that we have some comparison. And that's what we're going to use today to talk through a lot of the examples, is being able to compare one test against another.
Mm-hmm. If I'm talking to someone, an agronomist, a farmer, and they've got an area that, by all appearances, ought to be performing similarly, but it isn't, and they're scratching their heads about it, all right, it's a good opportunity to look for whether there might be something you could manage that you just haven't measured yet, some invisible biological root cause, right?
So what are we talking about testing? We're talking about biology. So we think about all the components in the soil and we look at biology, it makes up 0.5%. So a very small portion, but it does a lot of heavy lifting. And those items are water retention, nutrient cycling, disease prevention and suppression, along with abiotic tolerances and nutrient retention. So with that, Scott, you want to show us how we actually do that?
Well, it just so happens that my heart is 0.5% of my body weight, I estimate. So small, but essential, an essential driver and motor of the whole system operating. And what are we looking for as we test?
Yep. So in this example, you can see we've got some disease, we've got some nutrient deficiency, we've got a whole host of things. So it's really about using all the available resources that you have now and adding microbiology or soil biome testing to understanding what is really going on in your soils, and could we have prevented, or how do we mitigate these stresses or instances?
Mm-hmm. So this whole webinar is going to be about the answers that you can find in DNA that you won't find elsewhere when you have poor emergence, poor stand, stunted growth, signs of nutrient inadequacy. So brief overview of how bee crop testing works. What do we do? There are microbes living in soil, any soil. It doesn't matter what, even if you autoclave soil, it's very hard to get rid of some microbes. So they're always there. We extract their DNA. Then we have genes on genes on genes on genes. There are unbelievable amounts of DNA, of base pairs in any given tiny sample of soil. And it is not cost effective to sequence all of that. So what we do is we snip out just the piece we need to identify who it came from. Bacteria, archaea, fungi, they all have a marker gene that has a very low mutation rate. It's very consistent, and it serves as a fingerprint to identify the species or the genus that organism came from. So with those marker genes, we can identify the species, and then the magic of bee crop is the system of bioinformatics that we have then built on top of that, where we can say, we have this gene sequence. Here's its closest relative for whom we have a full reference genome on either end of the family tree, and then we can, with a known confidence, say, okay, well, this one is able to produce auxin. This one is able to nitrify, make nitrates available in a soluble form to the plant. These compete for nitrogen. They prefer nitrate as their favorite nitrogen source and will compete with the plant for it. These are able to suppress or kill fungal pathogens. And with that information, we compile that and report metrics, indicators of soil functional capacity in all of these different areas. Today, we're focusing on the middle: soil-borne diseases, biocontrol agents, plant growth hormone producers, and organisms that help plants adapt to stress. In the future, later this summer, we will have a webinar where we go in depth to the nitrogen, phosphorus, and potassium pathways to discuss how we can use these biological functional insights to inform our nutrient management, our fertilizer source, our application strategy. So please mark your calendars. I'll remind you again by the end, but we've built out that presentation and we're quite excited about delivering value with it. So by the end of this, we're going to be able to look at a comparison between two samples like this. Here we're looking at two samples. On the left, a sample that came from the poor performing ground. On the right, the sample that came from the strong crop that is right nearby, and by all accounts, seems like it ought to be doing as well, but they've been working on it for 4 years. It's chronically underperforming. So everything in aqua scored higher on the poor crop. Everything in purple indigo scored higher on the strong crop. We're going to circle back to this and break it down into what a simple pair of tests can teach us. There are, in fact, a whole lot more metrics than this. There are many bee crop indicators, but we're going to go through this. Basically, bear with us as we explain the science behind it, because for each chapter of this, we will get to a very simple decision-making heuristic to simplify this and help you make agronomic decisions. So we have 3 main chapters, looking at pathogens and biocontrollers, at stress and at growth hormones. So let us embark. To explain how it is that young crops experience their environment, I want to give the example, the analogy of a basset hound. A basset hound has an incredible sense of smell. And what its nose is doing is there's all these receptors in the nose for volatile organic compounds or compounds that it can interpret as an odor. And it will get, just thousands of molecules passing through its schnoz, and it will detect patterns, not necessarily just a single molecule, but patterns of molecules. Some of those signs will be irrelevant. It can ignore them. I'm smelling, azaleas, not important to a basset hound. But it may pick up patterns that are meaningful to it. For instance, friends. My owner, the one who feeds me, the one who trained me, the one that gives me shelter. It may pick up a smell of a duck or of a squirrel. And that is a prey opportunity, an opportunity to have some fun, get a reward from the owner. It can pick up opportunities like a mating opportunity, Mrs. Bassett, or rivals. It could pick up the smell of a rival. And so its nose you could think of as a pattern recognition receptor. These smells are patterns. They are odoriferous patterns. And the nose is the recognition receptor. Plants are covered with their own pattern recognition receptors. That is the formal scientific term. So a seed is sensing its environment. It has receptors on the cells around the outside of the seed. It is planning its time to germinate, and once it germinates, every root hair is coated, every plant cell is coated with pattern recognition receptors, sensing their environment, looking for smells, to use our analogy, that could indicate opportunities, threats, and in the case of pathogens, it's going to look for the odor, the stink of a pathogen. So here we have Fusarium, here we have Pythium. They each release molecules as they're going about their life in the soil, looking for a young crop to exploit. They release molecular patterns that are like molecular fingerprints of the pathogen. And so the plant picks that up with its pattern recognition receptors. And when it does, what that produces is pattern triggered immunity. That means the plant has smelled a threat and it is going to go ahead and take proactive steps to set up defenses. It's going to thicken its cell walls. It's going to turn on the alarm bell by sending defense response hormones like salicylic acid or jasmonic acid, depending on the pathogen, more often salicylic acid in our case. Throughout the plant, it's going to adjust its other growth hormones to prepare a response. It's going to produce reactive oxygen species, antifungal molecules in order to defend against the pathogen. And this leads to a trade-off in resource use called a growth defense trade-off. And what that means is basically there's two different places that a plant can put resources. That's a simplification, but it could put its resources into growth, or it can put resources into an immune response, into defense. There's a ceiling for the total number of resources the plant has access to, especially when it's young and it's living off of the seed. Any ounce and any smidgen of energy that's going into an immune defense is not going into growth. So here's one way of visualizing that. Top of the bar is 100% of the available resources. If it's not threatened, or if it just chooses to prioritize growth, you have a larger plant that is more vulnerable, less immune investment, or it could just prioritize immunity, small plant, well equipped to defend itself, but this may be a cause of stunting in the crop. Shout out to He Webster and He for those last two images. So thinking about our disease triangle, you have to have a pathogen and susceptible host and the favorable environment to end up with disease, full-on disease. But if you just have high abundance of a pathogen and it's producing those pathogen-associated molecular patterns, those PAMPs, that can lead to pattern-triggered immunity in the crop, even without having full-on infection, even without having disease. You can have a loss of yield potential without full-blown disease. So now, Mark, let us talk about how we take that concept and use it to uncover, to make decisions about whether this could possibly be an element that is yielding, producing poor growth early on. So we have a decision rectangle here, decision square here. On the left hand, we could have pathogen abundance that is high or low. And above, we can have a natural capacity of our soil to suppress or control or protect the plant from that pathogen, which is high, here's trichoderma, here's bacillus, or low. So if we have a soil with high potential for biocontrol and low abundance of pathogenic organisms, we have a disease suppressive soil. Yeah, pathogen abundance is not likely to cause poor crop performance. If the pathogen abundance ever increases, we already have the police force, we already have the guardsmen in place manning the battlements. If we have a low capacity for biocontrol and also a low abundance of disease organisms, it's free real estate. All right, there's no immediate risk, but if the pathogens, if the conditions get more favorable, if they figure out, hey, there's a plant here, we can start to exploit it, and the numbers increase, you don't have defense. The plant's going to have to fend for itself. So there's no immediate risk, but there may be risk later. in the case of lots of biocontrollers and a high number of pathogens, that soil is embattled. The pathogens are abundant, but the soil is capable of actively responding. So this could be a cause of poor crop growth, but if you're going to deal with it, you're not going to be able to improve, it's not for lack of biocontrol that the crop is facing this threat. And then worst case scenario, low potential for biocontrol, high disease risk, that soil is vulnerable. The disease is there, it is in force, it is coming for you, and it is unchecked. So as soon as conditions get right, or even now if the crop is sensing it and prepping an immune response, this could be drawing down on resources for growth. This is a likely contribution to poor performance. So Mark, I'm going to show you some reports, and you tell me which quadrant you're seeing them land in.
Okay.
This is a tomato crop.
So I see we have a couple pathogens that are showing very high. Looking below, we've got a few more that are in the low risk, but looks like we've got a fair amount of risk. So if we look at a couple of our biocontrol metrics, fungicide agents high, bactericide agents are high. I wish the others were higher, but at least those two are high. So I would say we've probably got a pretty decent soil or area within the field to
embattled soil. Disease is up, there's a high risk, but also the biocontrollers are there. Your soil is able to respond to the threat. Now we'll circle back to this in a minute as to whether this is enough or not.
Sure.
So here's another.
Looks like we've got another high pythium rot exposure and some other diseases that are available.
And it's indicated by the picture. This is an early season threat, and these are just little seedlings that are already suffering from it.
So now I see we've only got one side of the equation. The bactericide agents are high, but our fungicide is low. So, and everything else is low. So given this, we have some challenges.
And pythium as a fungal disease, your antibacterials aren't going to help you. And so here we have a vulnerable soil. We have an unchecked disease risk.
Yep.
And, it should be noted, we can't detect with our technology insects and nematodes. But if you're aware that you have repeating nematode populations on your soil, you're going to want to be seeing a higher capacity of your microbiome to combat repeating nematodes. If this is very low, okay, you have a vulnerable soil. So a word about biocontrols organisms. Here's our good friend Benjamin Franklin reminding us that an ounce of prevention is worth a pound of cure. When you are relying on biocontrol, it doesn't work reactively. You cannot apply trichoderma after your plant is already suffering from a disease and expect it to work. The biocontrollers have to get to the party first. So this is not the subject of the webinar. We just want to drop it in here because we're talking about biocontrollers. Top 3 considerations for effective biological disease control. Mark, will you take the wheel?
Sure. 1, target the biocontrol products that are specific to your pathogen target. It doesn't do good for applying a product A that only works on disease B. So make sure that we're matching up what you're trying to treat with the appropriate biocontrol or appropriate control agent. Again, we'll make sure that it will perform in those conditions. If you are applying something that maybe has a pH sensitivity and you have that pH sensitivity in your field, you're probably not going to get the response you're looking for out of that. So make sure that you are matching biocontrol or control agent that matches the fields. And the other part is treated seed or a drenched seeded strip to ensure that biocontrol or control agent is first to the party, that they have a chance to build up and be there rather than trying to react.
Very good. You wanted to nip it in the bud. So that's pathogens. We'll circle back to them with the final stage, but now let's look at stress. So Mark, what are we referring to when we talk about crop stress?
So stress typically we put into 2 categories. Biotic stress, so you've got the insects, bacteria, fungi, all the as the rabbits you see on the screen, or we have abiotic stresses. We're quite familiar with those, heat and drought stress. But also pollution, salinity, depending on where you are in the US, you could be facing salinity issues. But typically try and break those down into the abiotic and biotic stresses.
And you can add to this, lack of oxygen, mechanical stress from compaction, hail, herbicide stress would fall into this. Crops have to cope with all of these. And so the question then is yield potential. How much is lost? How much is salvageable, mitigable? How? Do I need to replant, treat? What's my urgency level? If I leave it, does it change my economic decisions about my next pass? What could I have done differently? Finally, if nothing can be done now, what could I have done differently to avoid this in a coming season? So I'm going to do a brief rundown of what stress adaptation metrics we report. Each one of these could be its own webinar, so forgive the brevity. But the first is exopolysaccharide production. Big word for a simple concept. So saccharide means sugar, poly means it's a chain of them, exo means it's going to be outside the cell. It's an outside cell chain sugar. What that looks like is, here's some images of bacteria. They want to stick themselves onto their home in soil so they're not washed away whenever there's a heavy rain. I love this image on the right where this bacterium is, just hanging on for dear life to the mineral particles around it and to organic matter around it. Now this is dried out. You have to dry out samples to get scanning electron microscope images like this. Usually this would be a full-on soup of exopolysaccharide around this bacterium. So essentially, here's our chains of sugars. They have these molecules, they have groups around them that are very attracted to water. They produce hydrogen bonds with water. There's van der Waals interactions. That creates a highly absorbent, water-holding environment in which organisms can communicate and seek resources. That's what gives you this lovely bloom of exopolysaccharides. What does that look like on a macro scale? It looks like soil aggregation. It looks like water aggregate stability. I love this video from Valent Biosciences you can find on YouTube, in which they have two soils, and the one submerged in water, it holds its shape, it holds together. That is exopolysaccharides in action. These rhizosheaths, the soil clinging to the root, creating a nutrient holding, protective, comfortable environment. That is all exopolysaccharides. Heavy metal resistance, microbes sequester heavy metals, they complex them in larger forms so they're not in a toxic form any longer. They precipitate metals out of solution so they can't be taken up by the roots. They transform them, they chelate them. That's all wrapped up in this heavy metal resistance category. Salt tolerance, microbes they help reestablish ion and osmotic homeostasis. They're far more, the ones that do this work, they're far more salt tolerant than plants. And so they can remove salts from solutions so the plants then don't have to deal with them. They also enable the plants to keep growing via hormonal regulation. Then you can choose how to pronounce this. I've heard siderophore, siderophore. The point is it's an iron chelating molecule. A chelator, all it is, it's a molecule that's designed to grab a large atom like iron. Siderophores are specialized for iron. Here's a little just line and stick drawing. You've got these 6 points that are going to encompass and help bind to the iron in different spots so that they can hug it, embrace it, peel it away from the mineral it's attached to. Here's how that looks in a ball and stick version. The iron is this dark blue in the middle, and it's been hugged away from the soil mineral substrate by these reds. And so that brings us to the hormonal help, ACC deaminase. ACC, it is a precursor to ethylene. Ethylene is alarm bells for your plant. So here's our ACC. I love this diagram. You've got a stressor. ACC is being produced. It can either turn into ethylene, in which case it will tell the plant, conditions are bad, alarm, shut down growth. Just chill out, ration your resources, wait for times to get better. And that will lead to less biomass, less photosynthesis, less protein content. That's a yield loss. Or it can be intercepted by ACC deaminase producing plant growth promoting microbes and not turn into ethylene. And therefore you don't get that growth shutdown response. So here's the source of this lovely image. Thank you, Sapri and co-authors. Salicylic acid is a plant stress response hormone. It responds to biotic and abiotic stresses. It can disrupt biofilm production. It can stop pathogens from being able to communicate and coordinate through disrupting quorum sensing. It can lead to an accumulation of reactive oxygen species. It is the molecule responsible for systemic acquired resistance in plants. So microbes can produce it to prime the plant before the plant has had to notice the pathogenic molecular pattern we saw. The microbes can prime the plant ahead of time. It's an intelligence network throughout the soil if you have microbes that can produce this. And then abscisic acid is a plant growth hormone that, watch the recording and pause if you want to run down a full list of the abscisic acid functions in the roots, where our soil microbes help guide root architecture to essentially improve water use efficiency. That intelligence network will tell the crop where water is, where it isn't, how water availability is changing. That will open and close stomates more effectively to make the most out of water resources if they are limiting. So that's what we indicate. That's what we measure with bee crop testing. Now we come to our decision grid. Stress level. You, the land manager, the agronomist, you know what the stress level is going to be. You know the ground. So you can have, let's take heat stress, drought stress. It can be high, it can be low. Now we look at our bee crop. What's our capacity to mitigate that stress? Be it salt, be it heat, be it drought, be it salinity, be it, sorry, I said salt, be it heavy metals. That can be high. Here's our microbe blowing a cooling fan on our plant, or it can be low. So if we have low stress and high capacity to deal with that stress, we are stress ready. We are primed. The microbiome is in position to support the crop when and if that stress ramps up. So best case scenario. If we have low stress and low capacity to mitigate stress, then this is irrelevant. There's no concern. Unless you expect that stressor to rise later, then you might want to push for a stress-ready soil. In the case that you have high stress adaptation and high stress, much like our last example, you have a stress-adapted soil, whereas your microbiome is ready to support the crop through this stress. To help it ride through that stress like there's no problem. If the stress is too much, then you can always have too much for the supports that you have, in which case, if you are going to improve adaptation to this stress, adding a microbial inoculant is not likely to help you. You already have those. So you're going to want to choose an abiotic stress adaptation product, say a protein hydrolysate, a seaweed extract, a targeted nutrition to help your crop's own response. These are just examples. And then finally, if you have high stress, you're going to have high stress here, or you do now, and you have a low capacity to mitigate it, your soil is in distress. You have no natural mitigation services. This is a case where inoculation with a stress mitigating microbe, a Pseudomonas, for instance, is more likely to be effective. Again, ensuring that your field conditions are hospitable to the microbe you're putting out there. And then address the root cause where possible. Address, can you armor the soil to lower the soil temperature? For instance. All right, Mark, here's a soil. Here's its capacity to adapt to stress. Let's say it's facing drought stress, heat stress. How well off is it?
Looking at the metrics that we're going to key on, I think if you start highlighting them, we're going to see the exopolysaccharides, ACCA deaminase, abscisic acid are all high to medium. This area of the field, we'll say, is ready to battle and handle some stress.
Very good. Room for improvement in the salicylic acids, but all in all, above average, relatively well off. The plant can recruit and lead to reproduce these that are present that it needs help with.
Again, not saying that it might not need some additional help, but at least from the initial onset, we are primed and ready to go.
Right. Biologically, well off. That doesn't mean, even if I'm healthy, I occasionally need the help of medicines, of vitamins, and so on to fight something off. So this is, depending on level of stress, this is either stress ready or stress adapted. I said it was stressed, so this is a stress adapted soil. All right, another example. What do you make of it?
I'm looking at this, Scott, and I'm seeing we've got challenges. So the only thing that's high is abscisic acid. All my others are in the very low or not even detected. Looking at the microbiome. So we have some challenges.
If this is an alkaline soil, I don't have the capacity to produce siderophores. I could be having some of that
Iron deficiency.
Iron deficiency, chlorosis in my soybeans, for instance, because you need chelators if you're in an alkaline condition and iron becomes unavailable. All right, abscisic acid is okay, but I'm really not satisfied with that. I have here a distressed soil. If I've got high stress, it's distressed. If it's low stress, then it's irrelevant, but we're saying this is a stressed soil. So
We need help.
Here we're jumping back to a way of visualizing two samples. This is a pair of samples that I looked at earlier this year that they were apparently quite similar soils, but you can see that biologically they had very different capacities to deal with stress. Global stress adaptation scores up 35 points. And then 2 out of the 3 plant growth hormones we measure were up substantially. Rarely do I see differences this large on soils that are ostensibly similar. So, which is a great segue to chapter 3, growth hormones. Plant growth hormones. If you look at a rhizosphere, if you look at the root zone, you will find microbes there who are plant growth promoting microorganisms, bacteria and fungi alike. Part of their whole survival strategy and fitness strategy is to partner up with plants and support the plants in exchange for sugars to feed themselves. So these guys are working hard, many of them, in order to produce, in order to build together auxin, cytokinin, gibberellin, salicylic acid, abscisic acid. We covered the last 2 of those in the last section, but auxin, cytokinin, and gibberellin are the big 3 plant growth hormones. These guide how the plant proliferates its root system. Is it going to grow down? Is it going to grow laterally? Is it going to put off, is it going to proliferate root hairs here, or is it going to power on and keep looking for a spot with greater nutrient richness? This is all guided by plant growth hormones. And if our intelligence network in our root zone can produce these to signal to the plant, there's a resource pocket here that you can exploit, give us some extra root hairs, or there's a lack of water here. These, A, they give our plant intelligence, and B, our plants, it is more expensive metabolically for our plant to build these than for our microbes to build it. The microbes are like, China in the 1990s building all of our cheap plastic goods because they could do it more cheaply. The plant is like the US in the 90s, or probably still today, that is outsourcing labor, outsourcing its work to its manufacturing to cheaper labor done by these microbes. So these 3, gibberellin, auxin, cytokinin, I want to point out that the growth stage is relevant to their importance. These 3 are most important during vegetative and reproductive growth. If you are testing in the fall after harvest, these 3 metrics won't mean the same thing than if you're testing during the active growing season. If you want more information specifically about each one of these, then check my LinkedIn page. There's a whole resource directory of resources that I've written out specifically about these, all the plant growth hormones and the microbiome and their relationships. So jumping straight into decision making, what's our growth stage? Are we in the active growth stage? Is our plant green, or are we in the off season? And then what's our capacity to produce plant growth hormones? Is it high? Is it low? It's our labor force. So if we have a strong labor force and it's out of season, man, that's a 1st round draft pick soil right there. That's MVP. It is ready in and out of season. If we have a low labor force and it's out of season, irrelevant, no concern, but potentially reason to check back during the growing season. A simple test to see if these populations boot up and grow when they're needed. If we have a high number of laborers in season, you've got a crop growth promoting soil. I forgot to mention this. Organisms that can produce plant growth hormones, that is a good proxy for just plant growth promotion in general. They tend to have multiple functions, multiple services they give, producing plant growth hormones being just one of them. So I use the plant growth hormone section of the report just as an indication of, do I have plant growth promoting microbes in my soil, period? Do I have friends? And in the case that we have a low labor force and it's in the growing season, man, your good guys are AWOL. You are missing out on opportunities to promote growth. It's not like, it's not lost yield, but it's lost yield potential because you could be growing more aggressively, more prolifically, more vigorously. So in this case, inoculation, cover crops, improving the soil conditions so that your plant growth promoters can live. This is the case too, where if you have I know that some companies have within their portfolio plant growth regulators that include indole-3 acetic acid, canadenine, gibberellic acid. This is the case where you may get value out of those, may be worth trying. But in the other cases, not. So let's put it all together, circling back to that initial comparison, this initial case study. This is looking at irrigated ground in the southeast, where chronically underperforming ground, they had treated it to try to improve the nutrition on that ground for years and years, but it has not been responding. And this year again, it is just from the very early going, it's underperforming. So Mark, what do you see? What jumps out at you first, looking at the most salient differences between these two soils?
I see our fungicide agents are up, zinc transport's high, functionality's high, and my exopolysaccharides. So we talked about that. Those are all. That's in the strong crop.
And so in our poorer performing crop, what are the largest in higher scores?
We've got diseases, and we have a global nitrogen score that's low, and our calcium transport's low. So some pretty important cycles.
These are
those are on the high side. I'm sorry, you're correct. Those are on the high side.
So notably, one of our most important metrics for vigorous growth, global nitrogen, is higher in the poor performing crop. Matter of fact, the soil chemistry tests off this field also showed the nutrition, the available nutrients, better off where the crop is worse off. Across the board, nutrition was better off. The cycles and the available nutrients better off on the poor performing side. And so it's very likely these disease issues are a problem, because if we have much higher disease risk, the pathogen associated molecular patterns are there, but our capacity to deal with them is not. We had a much lower fungal control score on that ground. Actually, we know it's higher on the good ground, but let's just check and see if the absolute score is bad off. Okay, in the poor crop, it's in the very low bracket. Okay, we got the diseases, we don't have the controllers, vulnerable soil. The crop's having to respond by preparing its own immune response. In terms of stress, it was lighter soil, faces more moisture variability. If anything, I'd want to see higher stress adaptation on the left-hand side here. But no, if anything, I see it on the right-hand side. They're not that different, but the exopolysaccharides are significantly higher on the heavier ground, where they're arguably not quite as essential. And there, yeah, okay, very low bracket for the exopolysaccharide production in the poor crop.
Again, can you go back a slide, Scott? Again, we see that we have one sample in the very low and one in the medium. Again, there's still room for improvement, but that is a considerable difference in performance and how crop response could occur by just having that 26-point difference between the low sample and the high sample. And again, we were talking about the decision square of, do we have the capacity, but might there be the need for additional help? This is where there's a lot of products out there. If it is a stress or something that we can work with, this is using the information of how, where, and when might be the best use of those types of products in conjunction with knowing where you stand and how widespread this challenge could be.
Mm-hmm. And there are occasions where, jumping back to all the stress metrics, stress and phytohormone, there are occasions where I see the highest stress adaptation scores on the highest stress ground. I had a project earlier this year where this was a fully mapped version. So we're looking at a B-crop test report here, but B-crop farm is where you map these metrics across the ground on a per acre basis, 10 by 10 meter resolution. And on this particular farm, the global stress adaptation score perfectly matched all of their lighter, sandier soils. And so there are cases where what you have is not a distressed soil, but a stress-ready soil, and it's maxed out. That's where you use an abiotic stress adaptation or change your nutrient management to improve stress adaptation. We didn't talk about these metrics, but it's notable that overall soil health, functionality, biodiversity, soil quality, they were higher on the strong crop side. This is indicating to me that soil health across the board can be improved on the poor ground and lift multiple boats in terms of the different functional indicators. So, in summary, bref, we have talked about, Mark, will you read these
Yeah,
off?
so I think one of the key points here is we talked about is pathogens do not have to infect to draw down yield potential. Again, if they're getting those signals and they're expending energy to fight off a pathogen or to prepare themselves to fight off a pathogen or to deal with a stress or whatever the challenge is, you think about that crop, if it's expending energy in that direction, it is not expending energy in the direction of yield.
Mm-hmm. And it should be noted, of course, that your pathogen risks early in the season are also forecasting where you may have actual disease and infection later on. Another project in Virginia this year, Bee Crop Farm was showing high risk of multiple early season diseases. We had a wet spring, very wet spring, like much of the Southeast and all up and down the East Coast did, maybe elsewhere, but those are the areas I'm most familiar with. And the recommendation that Bee Crop Farm had put forward was a trichoderma. Just given the logistics of how we, how quickly our partners on the field there, our client on the field was able to get their samples pulled, they didn't have time to buy the product in before they needed to plant. And we had that wet spring. They have had cotton seeding disease complex like we haven't in recent history. And so I'll just put a plug in here for testing disease risk early enough or even in the fall to plan ahead decisions around planting, even rotations and genetics.
So number 2, natural biocontrol agents shield that crop, but require proactivity. Again, if you're looking at using one of these products, it's hard to put a seed treatment on after the seed's already been planted. So making sure that if we're looking at what our plan is and making sure that we've got the adequate resourcing and things in place to make sure that it is put in and has the chance to work to the full benefit that you need for the problem you're trying to solve. Comparative DNA samples. We've talked about this. This is the power of using 2. Imagine where we would be, Scott, if we only had 1 sample to look at. We were trying to make the assumptions and off of the good sample or off of the poor sample. Again, I think more is better. Depending on what you're looking at. 2, 3, 4 samples. Being able to look at what is going on in your field. I think we've shown just from the categories we've talked about that yeah, some need to be taken care of up front, but there's also a host of products and tools out there that growers have access to today to help mitigate stress, whether that's heat stress, drought stress, some of the abiotic stresses, or even dealing with biotic stresses. So
imagine I take just 1 sample from the poor performing ground and it tells me my global phosphorus and global potassium scores are 33 and 26. I might assume that's the problem, but if I picked a sample from my strong ground that I'm happy with, and it tells me my global phosphorus potassium scores are 35 and 23, okay, then that's not what's causing this poor performance, because my strong ground already has comparable scores. I need to be making comparison to figure out what's different between the ground I'm happy with and the ground I'm unhappy with.
Yep. Number 4, plant hormone producers reflect wider growth promoting services, but seasonality is relevant. Sorry. So soil DNA can enable you to pinpoint biological root causes of crop performance. I think that's then the whole point of talking through this, of looking at what can we do from a proactive and also reactive of using soil biology to understand, when, where, how, and why are we seeing areas of the field. I'm sure you can drive past fields. I live in central Indiana. We had our challenges with water this spring, but we also have areas that same topography and areas of corn and bean fields that look very different. from a landscape position and soil type and those types of things are looking good. But understanding what's actually happening from the biological level of what's happening in the soil that is driving some of these responses, and then what are our appropriate recommendations and actions that can be taken once we have access to this type of knowledge.
Mm-hmm. Emphasis on you. Soil agility can enable you, thanks to the legwork that our development team has put into making this, taking a string of letters or a list of indecipherable Latin species names to here's what your soil can do. And then just screen cap, hop on YouTube after this is posted, screen cap those decision grids, and you can use soil DNA to make relevant decisions, take action for your crop, for your client's crops. And so today we covered the center section. Using the data you get, say, in early season, you can forecast potential nutrient cycling bottlenecks later on. I mean, until V3, V4, the plant's living off the seed, but you can go ahead and forecast. I'm kind of expecting these blockages later in the season. You might adjust your nutrient management for the remainder of the season, for your next pass or so, with that information. This is important in and out of the growing season. And so again, August 27th, mark your calendars. This is going to be a useful presentation, breaking ground we haven't really broken publicly in a webinar before. Mark, do you want to say anything more about that?
No, just some new concepts. Yeah, I'll probably we'll tease it that way. We're going to introduce a new concept that I think bring a lot more value and understanding of the benefits of really understanding mineralization and tie up or immobilization in the soil. So if you can, please make sure once we get the information out, sign up and register for that August 27th webinar, because I think it's going to be a doozy. We've already got it planned out and looking at it, and very excited for that date coming up.
Huzzah. Okay, we welcome you to test with us to get insights on your own soil. So Mark, will you discuss these two methods, two approaches?
Yeah, so the information today that you saw, we offer two platforms. So go check out biomakers.com/bcroptest or biomakers.com/bcropfarm. So the data that we've been looking through today was based off of using bcroptest. We've got a web portal that you can look at your results and look at all those charts and graphs and information, and use that to help decipher how well, what your challenges are. Use it for predicting. If you're running these types of tests and you're running them on multiple growers, you can better scale, how much of a mitigating product, stress mitigator am I going to need? What products should we be taking a look at for the next time this crop is in the field? If you want a higher resolution look at that information, then we have our precision platform of being able to look at, and that's bcropfarm. We run it through a proprietary analysis to determine where those samples need to be pulled, and then go out, pull those samples, bring it in our platform, and generate a lot of the maps that you saw on either side of the slides today, so that you get a visual representation of where and what scale or of challenges I'm having. Is most of my field having one of these challenges are open to a heat or drought stress? Are there areas of the field that are going to be better able to handle that? Does that affect my timing? So if I'm looking at the weather and I'm using weather and scouting, can I now use this information to better predict which fields do I need to target first? Timing can be mission critical when looking at handling stress or handling any of these challenges. So where am I going to pool or put my resources first? And then to what degree am I going to apply those resources? Is it the whole field, half the field? Do I need to run a variable rate prescription to address these areas of the field, treat certain areas first, and then manage, monitor, and make a decision to treat again? There's a whole host of options to managing products as well as ROI, depending on what scale and where you feel comfortable jumping in and utilizing the technology.
All right, that's all the content. We got through it, Mark. Virtual high
Scott,
five.
we did it. Happy day. We made it.
We have 11 minutes for Q&A.
Let's see. So I'm looking at a couple of the questions coming in, and I think you may have answered. So from your trials, have you seen biocontrol products be more effective when the soil has low stress adaptation?
Have we seen biocontrol products be more effective when the soil has low stress adaptation? I can ask our agronomy team if they have observations about that. I don't think my experience of tracking, often, I'll put it this way, often clients take our report and then they don't necessarily come back and tell me, oh, we added this biocontroller, here's how we responded. If we have a subsequent conversation later in the season or the next season, I usually have notes and ask them about that, but I haven't had enough instances where someone said, I had these results and I applied this biocontroller and here's what I observed. So please, you try that and inform me so that I can answer this question expertly next time someone brings it up.
I'm looking through. Unfortunately, I'm having some technical difficulties at the moment, Scott, on my other system we're using.
Okay, let's see. Do you have plans to add more than NPK nutrient cycling microorganisms to your assessments? Done and done. As a matter of fact, carbon, nitrogen, phosphorus, potassium, sulfur, copper, calcium, magnesium, iron, manganese. Am I going to get them all? Chlorine. 8 of the micronutrients beyond CNPK are a part of the report already, which is more than you'll find from any other soil DNA test that I'm aware of. So great question. Will you be adding higher level microorganisms like protists, nematodes, etc.? While having information on bacteria, fungi, and archaea is very important, it only tells part of the story. It would be awesome to have information on other organisms to make up a functioning soil food web. I 100% agree. On the other hand, if you're used to looking at samples through the microscope, you'll know from commercial agriculture how rare it is to even see those in a sample. And so we've had to ask the question, is it relevant for us to add $50, $80, $100 to the cost of a sample in order to 9 times out of 10 say none present, none present, none present? We've decided that hasn't been the right choice to make at the moment. But what that means is if you can invest in a microscope or have someone who has a microscope, soil DNA to cover the bacteria and fungi that are low resolution on a microscope to know what they do, with a microscopy assessment to see the higher level, predatory organisms, great complement. I think the soil food web approach is a great complement to biomakers and vice versa. Maybe someday.
Scott, I finally got my thing back up. There's a question I'd like to address. It says, I did not see your technology being used in Colombia, South America. How can we get your tests here? Actually, we have done a fair amount of testing in Colombia and South America. We've got a team that's working down there. I would say reach out to myself afterwards, and I can put you in contact with the right team member that will be able to handle your needs, because we do have labs, and we have the ability to get samples from all over and bring them to our Davis lab in California for DNA analysis if we don't have a closer lab in country. Again, we have several lab partners, but yeah, just reach out to me. And I will make sure that we get you put in contact with the right team member to handle your needs.
Excellent. I see 2 questions here I want to take. 1, how does a customer request a comparison of soils in the B crop results? I've only received them as separate reports. And I tell you what, even if you have 2 reports, but especially if you have 4, 5, 6, 10, 20, it is a pain to have a whole bunch of PDFs out and try to glance back and forth. So yes, absolutely. It's available to you in the B crop portal where you'll get your reports. On the left side, there's a button called Toolbox. The comparisons we were using are under the metric box plot. Click that. You can put 2 samples to compare. You can put a cluster of samples you can replicate. You can look at the samples from the bottomland on your farm and the upland on your farm. You can compare any cluster of samples and get those visualizations we looked at. The Toolbox is a underappreciated resource. I use it daily in the course of delivering results, and it's free to anyone who takes even a single B crop test. And the other one I love here, you report results that are based on function. You report functional results while you analyze the microbial species based on their DNA. Why tie this together? Why not also report the microbial communities responsible as well as those that are deficient? So the answer to this question, I had this question when I started this role. Over the course of over 100,000 samples, 10 years of work, what we have essentially concluded is, in line with the study of the human gut microbiome, the functional capacity of the community is more important to the crop outcome, to the ecological outcome, than the specific microbes present. There are dozens, hundreds of microbes that can perform any given function. And so you can have two samples with virtually zero overlap in the names of the particular organisms, while the functional profile can be very similar. My gut and the gut of a woman in Kazakhstan can have no overlap in terms of species, and yet they have to do the same jobs. They have to digest carbohydrates, proteins, lipids. They have to perform the same jobs. And so we don't report the microorganisms specifically, one, because it's confusing to most users. If you want your list of microbes, you just write our customer success team and say, I'd like the spreadsheet with my taxonomic abundances. You can have all your microbes and the relative abundances. What we don't do publicly is connect every single dot between microbe name and functions that they contribute to. One, because, well, then what's to stop someone from rebuilding bee crop from just taking some samples from us? And two, because it's actually a false notion to think you can do that, because so many of the species in soil don't have specific names yet. So we might class them to a genus, and if we can't class them to a genus to a family, and we can know something about their role and their functions, but you're not going to even be able to get that just from the names of the organisms. Something like 3 to 5% have names, and yet by using the family tree, leveraging the family tree concept, we can still pair each DNA sequence with a family, with a genus, with closely related species to produce the functional report. So short version is the function is more important to the actual outcome you have in the field, in the crop.
So Scott, there's one question I'll take in here of have you combined this technology with Red Edge tech deployed during the growing season? I think that's a fine idea to use. I think there's a whole host of metrics, whether it's straight NDVI, Red Edge, all the derivations of the imagery layers. I think those are fine ways to define areas to go sample. However, like I said, is we have bee crop test and bee crop farm, and bee crop farm we use over 1,000 different layers through our AI model, and some of those include imagery as well to define where are the best areas to look at that biodiversity to give us and be able to generate the maps that we generate. So I think if you're looking at just running bee crop tests rather than the full-on bee crop farm, I think the Red Edge or whatever metric that you find best to compare like areas or to discern the differences in areas, I think that's a fine choice to do. So Scott, it looks like we are closing in on time. Any last closing comments before we say thank you again to all of you for taking time out of your busy day? Hopefully this was beneficial and you enjoyed your hour of time with us. And yeah, Scott, any closing comments from you?
I most enjoy, maybe you can tell from the way I deliver the webinar, I most enjoy the ecology of soil and looking through results and helping people piece together what they mean. So I'm just hoping to hear from some of you who are ready to test and get some of these insights for yourself. So thank you for your time and attention.
Yeah, thank you for your time. Hopefully we've shown you some different ways to utilize this technology to incorporate into whether you're planning, your spraying, treating, whatever the process is you're looking to improve, but incorporating a new level of technology. Again, I know you've probably used soil testing or tissue testing or sap testing and all. There's a whole host of avenues, but now we have the ability to dig deeper, if you will, into what's actually happening in the soil and driving a lot of these pathways and properties that that are being expressed in the crop that you're growing. So again, thank you for your time. Thank you for your support. And Scott, you got one last word?
You gave me a far better closing comment. That case study we looked at, they spent 4 years trying to, grease the squeaky wheel, but with the wrong grease, addressing the wrong problems. My final word should have been, if you have chronically underperforming ground, let's help you try to figure out potential root causes, and not invest energy and sweat and resources into not the problem that needs addressed.
Yep. All right. Thank you again. We look forward to seeing you. We've got a couple more webinars, but hopefully we'll see you again on August 27th. So thank you again for everybody's time, and everybody have a great, safe day and rest of the growing season. Thank you.